So distinguished ladies and gentlemen, and Claire, I would like to welcome you today to this MENDEL lecture, which is given by Professor Greg Hannon from CRUK in Great Britain. And it's really an honour and a privilege to have him here, considering he's one of the main people who have made many seminal contributions to the field of RNAi, which many of us now work on. Briefly, because if I really said everything that was in his CV, he would have no time to give a talk. So I'm going to give a very, very brief overview. He's from the US, from Pennsylvania, and he did his bachelor's in Case Western Reserve University in Cleveland. He stayed there to do a PhD with Tim Nielsen, who's known for his work on RNA. After that, he moved to Cold Spring Harbor, and he was doing a postdoc with David Beach. And he remained there and became an independent investigator in 1999. And that was just around the time when RNAi began. So he then began to work on that and had a very successful lab for over 23 years in Cold Spring Harbor. He had won many awards. He was a Howard Hughes professor at Cold Spring Harbor. And then in 2014, he came to Cambridge and began working in CRUK, and is now and will be the new director of it from next February. What is amazing about Craig, looking at his CV, is like 10 years ago, because this is a real old one I got off the internet, he had 162 papers. So I have no idea how many he currently has. But I think what is more impressive is the number of people and PIs who have come to your lab, and what a great mentor he has been for the next generation. And that is really very, very impressive. So thank you very much for accepting our invitation to come and give the MENDEL lecture.
on my Okay, so what we're more interested in is the genetic and phenotypic evolution diversification that happens when a single founding tumor cell grows and elaborates into the complexity of cells that form a primary tumor, and then even more so as just a few cells from that primary tumor leave that site and form secondary colonies as metastases in other organs. Now, tumor heterogeneity is a relatively hot topic, and I think especially now with the advent of single-cell sequencing, many people are in the business of cataloging the variation in tumors. And I'll just highlight one of the very early studies in this area from a former colleague of mine, Mike Wigler at Cold Spring Harbor, who looked at reconstructing clonal lineages based on copy number variation in single cells from breast cancers. And I highlight this to point out that most of these single-cell studies are retrospective. And so they catalog the variation within tumors, and they catalog the evolution of cells within that cancer. But they say nothing about what that clonal diversification means for tumor development. And so what we're really interested in is not simply what heterogeneity exists or what lineages are evolved, but in what the different capabilities of those lineages are and how they contribute, how they influence the course of disease. So we wanted to try to study this question to understand the contribution of phenotypic heterogeneity to tumor development using a simplified model. And so we actually asked whether this was a process that we could possibly study using cell lines that had been established and grown in culture for many years. And in this case, we turned to a particular mouse breast cancer model, 4T1 cells, which was derived from a spontaneously arising mouse mammary tumor about 25 or so years ago. And this model actually has a number of different advantages. So it can be syngeneically transplanted into immunocompetent mice, and that, of course, makes it interesting for studying tumor host interactions. Or it can be transplanted into immunocompromised recipients. And so to ask whether we could study this process or this problem of tumor heterogeneity using this cell line model, we did a really simple experiment, which was to take the 4T1 cell line, to individuate cells within that simply by infecting a population of cells with a retroviral barcode library. We then bottlenecked that population so it had 100,000 founders, so that each of those founders was associated with a unique molecular barcode. We could then track what those cells did once we transplanted them into the orthotopic site of a recipient animal. And so we transplanted these. We let the tumors grow for a few weeks, and then we harvested not only the primary tumor, the tumor at the primary site, but we also harvested cells from all of these secondary locations. And so the question was, did any of these cells in this initial founding population show any differences in a capacity to colonize and occupy these secondary sites? So what I'm showing you here is just a heat map of sequencing, of quantification of the representation of those individual barcodes and samples that we recovered from mice. So each column is a replicate mouse, and there are, I think, 5 replicates in this experiment. And each row is an individual. It represents the sort of frequency with which we saw an individual founding clone in each of our samples. Now, we transplanted 100,000 founders into each of these animals. You're only seeing about 1,000 different clonal lineages here. And the reason is because we've only shown the clonal lineages that we detected in each of the 5 replicates, right? So this is so that we can show you the consistency of their behavior. Now, what was immediately apparent was that we saw actually, many of the transplanted clones contributed more or less to the primary tumor. But as we started to move into secondary sites, it was really only a subset of the cells that we introduced in the engrafting population that showed up, for example, as circulating tumor cells, and an even smaller subset of those that showed up as metastases at the secondary site. And in fact, if you look, a lot of the cells that you see in these populations at secondary sites are not particularly abundant in the primary tumor. Now, at that time, we couldn't reach back into that founding population, and for example, isolate, these particular barcoded clonal lineages and then study them. So we had to construct, sort of a simplified model. And so I did what, sort of every PI who's been out of the lab for a lot of years would do. I sent Simon and Elvin off, and I said, fact sort about 1,000 clones, grow them up clonally, mix them back together, and reconstruct a kind of defined model that we can work with. And so, my original concept of 1,000 clones ultimately turned into what we call a limited complexity model with 23 founding clonal populations. And I was actually a bit pessimistic that this was going to recapitulate the behavior that we'd seen with the more complex populations. But if it didn't, this would be a very short talk. Okay, so what we did was we took these individually fact-sorted clones, we mixed them back together, we pooled them to create this sort of limited complexity heterogeneity model. Again, introduced this at the orthotopic site and just redid the experiment that I showed you before. And actually, somewhat gratifyingly, we saw the sort of same behaviors in even this limited complexity model that we did in the much more complete one. And so if we just look at tumors at the primary site, we found that very early on, 2 weeks after engraftment, there were a few clones that dominated the primary tumor. And you could actually completely predict this behavior if you just asked which of these clonal populations proliferated most quickly in culture. So you looked at growth rates in 2D and 3D, and those were the clones. The ones that grew fast were the ones that populated the primary tumor at an early time point. But actually, if we just let it grow 10 more days, these rapidly growing clones are being lost, and they're being replaced by other clones, which seem much more able to contribute to the primary tumor long term. Now, we don't know the basis of this. We haven't yet studied that. But what we have focused on is the properties of these clonal lineages that give them preferential ability to colonize or to actually contribute to circulating tumor cell populations in the blood or to colonize secondary sites. Now, what I want you to notice here is that these CTC proficient and metastasis proficient clones are actually minor populations in the primary tumor. So it's really only a small fraction of the clones that contribute to the primary that can actually then leave the primary site and contribute to disease progression. So I want to first focus on what the properties are that enable these clones to be proficient to contribute to circulating tumor cells, and then later I'll focus on what it is that discriminates this pro-metastatic, this ability to actually colonize and create tumors at secondary sites. So we started by doing kind of and I'm going to kind of gloss over this bit because it's published and you can go back and look it up if you're really interested. But so we sort of did a divide and conquer experiment where, so we know metastasis, and this is a greatly simplified view, is a fairly complex process which involves the ability of that primary tumor to enter the bloodstream. So it's got to leave the primary site, enter the bloodstream, survive in blood, ultimately to create metastases. It's got to leave the circulation and actually proliferate in this sort of hostile, this sort of unnatural environment. So to ask sort of which of these steps was creating proficiency for contributing to circulating tumor cell populations, we just did an experiment where we compared animals where we introduced this sort of tumor heterogeneity model at the orthotopic site versus directly into the bloodstream. And so we did this by either tail vein or by intracardiac injection. But what we saw is, if you introduce it to the orthotopic site, you see this very strong dominance of only a couple of clonal lineages in circulating tumor cells. But that phenotype is completely suppressed by intracardiac injection. And so what that means is that it's not a differential ability of these cells to survive in the blood that makes the difference. It's really what makes the difference is their ability to leave that primary site and enter the circulation. We have other data for this but I don't have time to show you. So if we think about, what happens when those cells leave the primary site, there is at least a notion that the cells which are initially more epithelial might become more mesenchymal. They'll invade through basement membranes. They'll invade through the endothelium, enter the bloodstream. And so that sort of led us to think about, well, what does the vasculature look like, and what do the cells look like as maybe they're invading that vasculature? Now, again, I got to skip a whole lot of data because I have a few things that I want to talk about today. But one of the things that we looked at was how leaky the vasculature was. And you do this by just injecting high molecular weight dextrans that are fluorescent and looking at how easily they exit the vessels in the tumor and become lodged in the tissue. And so what we saw was that if we form tumors exclusively with those clones that had a high proficiency for forming circulating tumor cells, clones E and T, right? Clever designation, I guess, that the vasculature in those tumors was incredibly leaky. So we sent this to a pathologist, and we said, well, tell us what it is about that vascular that's different. Now, these are horrible pictures, and so I'll just kind of walk you through them. So here's a normal vessel. And you can see it stains with two things. So periodic acid Schiff, which kind of stains this sort of extracellular matrix that surrounds vessels. But importantly, it also stains with CD31, which is an endothelial cell marker. Now, if you look at this leaky vasculature formed by these CTC-proficient tumor cells, you can see that there is periodic acid Schiff staining. This is just a trace of the vessels. But they're not CD31 positive. So those vessels are not being formed by things with canonical endothelial cell markers. What they were positive for was M-cherry. And what's interesting about that is M-cherry is the fluorescent protein that we use to mark the tumor cells that we engrafted into the animals. So these vessels were actually being formed by the tumor cells themselves, organizing into vascular-like structures, which then plug directly into the normal vasculature, because others have shown that similar structures detected previously shows fluid flow by Doppler sonography in concert with the heartbeat. And so this is a process that had been discovered about 10 years ago. Was initially, let's say, quite controversial. In fact, even until very recently, quite controversial. Something called vascular mimicry or vasculogenic mimicry. Now, we all know that tumors have to recruit a blood supply. They have to be perfused. They have to have nutrients and oxygen, etc. The way we normally think about them doing this is angiogenesis, where tumors secrete factors that effectively attract endothelia to form vessels within the mass, right? But in our case, as I said, we see these vessels, these tubes, being formed by sort of this spontaneous remodeling of tumor cells themselves. And in fact, within the complex heterogeneity model, it's really just a subset of tumor cells that will form these structures. And what I can tell you is that if you stain just for those two CTC-proficient clones, they're not uniformly distributed throughout the tumor. They actually form these tubular structures, or they cluster around them. And we're not really sure what the sort of basis of that sort of morphological organization is. But one important point that I want to make here is, we all think about sort of metastasis being the lethal event generally in patients that succumb to cancer. But all of the things that people have described as being pro-metastatic, none of these actually confer an advantage, a growth advantage on the primary tumor. To me, at least, until somebody in the audience corrects me, this is the one exception that I know of where there could be positive selection, a benefit to the primary tumor for a process that promotes metastasis. And the way that we think this works, essentially, is that the cells that are capable of this vasculogenic mimicry have direct access to the bloodstream. All they have to do is shed from the inside of these vessels, and essentially they've entered the vasculature. We haven't proven that that's the case, but we very much think it is. So it's become increasingly appreciated over about the last 5 years that the presence of these vascular mimics is a marker of poor prognosis. And if you look across many different tumor types, and that we're working in breast, it's actually not even the one where vascular mimicry is either most prevalent or actually provides the worst prognostic marker. But across many different tumor types, the presence of these vasculogenic mimics is a marker of poor outcome, and you're about twice as likely to die of a number of different kinds of cancer if you have tumor cells that are capable of undergoing this transition. So as I said, we want to understand, how these clonally restricted phenotypic differences promote the progression of disease. And, vascular mimicry is certainly one such instance. But now we want to dig deeper and say, well, what's the mechanism that drives the ability of that clone to undergo this process, what we're calling the epithelial to endothelial transition? And that'll be important later because you want to think about this as plasticity, right? And so we did, sort of all the standard kind of omics things, RNA-seq, whole genome-seq, DNA methylation profiles, etc., comparing each one of these clones and then looking for those things which showed increased expression in the clones that were CTC proficient, clones ENT. And so then we took this is kind of a short list of the top differentially expressed clones. Here's lung mets, here's the two CTC proficient clones, and here's the parental line. And so we took these most differentially expressed genes. We then correlated those with patient data sets looking at expression in the primary tumors of patients which either would or would not later relapse with lung metastases. And two genes stood out, SERPINE2 and SLIPI, both annotated as secreted protein serine protease inhibitors. And so those were the two that were most, amongst these differentially expressed genes, the two that were most predictive of whether a patient would have a later lung relapse. So we chose to kind of focus on these, at least initially, and we could show that these two proteins individually were drivers of this vascular mimicry phenotype, because if we took a CTC proficient clone and we used shRNAs to knock down either SERPINE2 or SLIPI, we saw reduced, sorry you can't read that, but this is the number of vascular mimetic channels that we see in a tumor formed in vivo at the orthotopic site. And so we see reduced vascular mimicry if we take out either of these proteins. But we can also do the reverse experiment where we take poorly metastatic human breast cancer cell lines and we enforce the expression of either SERPINE2 or SLIPI. And in both cases, in these two examples, we see an increase in vascular mimetics in tumors formed from these cells in immunocompromised mice. And this correlates also with an increase in the metastatic potential of these otherwise relatively poorly metastatic models. So these two proteins seem capable of driving this epithelial to endothelial transition in vivo and of promoting metastasis. But what we also noticed as we started to try to dive a bit more deeply into mechanism, that those clones that were CTC proficient in vivo were actually able to self-organize into tube-like structures in vitro. So if we simply plate these in a semi-solid matrix, in this case Matrigel, these two clones, these are the CTC proficient ones, make these tube-like structures spontaneously. You see very little of that in the parental 4T1 population, and these are two non-CTC proficient subclones. This happens to be one that really dominates the primary tumor at early time points. And this is just for comparison, kind of a professional tube-forming cell. These are human umbilical vein endothelial cells. So we see this kind of vascular development property in vivo. It depends on SERPINE2 and SLIPI. We see tube-forming potential in vitro. Are these two related? Well, we think they are, because if we knock down either SERPINE2 or SLIPI, we inhibit the ability of these CTC proficient cells to form tubes in vitro. So we think we have an in vitro surrogate assay for this VM capacity that will let us much more rapidly progress both in the genetics and biochemistry to try to understand mechanism. And I don't have all the answers for you today, but I'll sort of update you on where I think we are. So SERPINE2 and SLIPI are secreted proteins, But what we noticed is that if we looked at the list of differentially expressed genes between clones E and T and then the non-CTC proficient, the non-VM proficient clones, there was a very strong enrichment for trafficked proteins, either secreted proteins or proteins that lodged in the plasma membrane. And so this started to make us think that there might be a more general secretory program that drives this process. And in fact, if all you do is take these VM proficient cells and you change the media a few hours prior to plating on the semi-solid matrix, so you deprive them of the conditioned media, of extracellular factors, the conditioned media, you greatly reduce the ability of these cells to form tubes in 3D. So we began thinking a little bit about secretory programs in general, and what we know is that one common property of highly secretory cells is that they, to one extent or another, activate the ER stress response. And the ER stress response has three arms. There's the PERK arm, the ATF6 arm. You can ignore those. I don't have time to go through all of the data, but they're completely irrelevant to this process, at least for now. But there's another arm, this IRE1 arm. Now, IRE1 is a kinase and a nuclease. The function of the kinase domain is to auto-regulate the nuclease activity of this protein. It gets activated under conditions of ER stress, and it uses its nuclease domain in a process called RID to directly cleave the mRNAs of secreted proteins. And so it also regulates the splicing of this gene XBP1, which then increases the expression of sort of chaperones quality control proteins. So this arm of the IRE1 pathway is irrelevant to what I'm telling you, but this RID arm seems actually to be quite important. And so the evidence for this is that we can manipulate the RID arm of the IRE1 component of the ER stress response in a number of different ways. There are a number of tool compounds that people have developed that inhibit the activity of IRE1, and we can also modulate IRE1 genetically. And if we do that, if we inhibit IRE1, again, reducing the ER stress response, increasing potentially the expression of secreted factors, we increase the vascular mimicry, at least as measured by our in vitro assay. If we do the opposite, if we agonize, if we increase the ER stress response by treating cells with tunicamycin, we can repress the in vitro surrogate for VM. So we can change vascular mimicry by tuning the ER stress response. Now this correlates with changes in either steady state SLP mRNA levels, or in fact, if we do measurements of RNA half-life, this correlates with changes in the stability of the mRNA of SLP, this secreted protein that we identified as a VM driver. But although this correlation holds, I don't think this is really the only secreted protein that drives this process, because we've been working our way down that list of differentially expressed genes, and of the top 10, 4 of those secreted or trafficked proteins actually contribute to this EET, to this VM forming proficiency. And in fact, one of those proteins is actually annotated in a truly crappy paper as a cellular receptor for SLP. Now, we don't know if that's true. I mean, the paper is really, it would be difficult to take that as evidence, but it does seem possible as we've annotated these two things as drivers of this process in a completely unbiased way. So cells care about the ER stress response for VM forming potential in vitro. VM is important for metastasis in our mouse model. I mean, do we really care about this with respect to human patients? And I think that we do, because if we take, in this case, the cell type or the breast cancer type that is the 4T1 model is most representative of, sort of a basal breast cancer cell type, and we stratify patients by whether they have high or low IRE1 expression. Those that have low IRE1 expression, again, lower ER stress response, potentially higher VM potential, they're about twice as likely to die from the disease as are those with high IRE1, high ER stress capacity. So it looks like, at least in terms of global ER stress, there's a correlation with outcome in patients. And we can also, in our model, manipulate metastasis by manipulating IRE1 and the ER stress response. In this case, we can knock down IRE1 with either of two shRNAs. We don't see an impact, a very big impact at the primary site. Tumors still grow, but we do see that this increases the ability of these cells to form secondary metastases. So what Ian then did was he said, okay, well, if I can agonize or antagonize VM and ER stress with either these genetic or chemical interventions, can I sort of create a gene signature that's representative of VM capacity? And so what he did was actually quite simple, which was to just essentially do expression profiling either in the presence of IRE1 inhibition or ER stress agonism with tunicamycin, took those gene expression profiles, created a pro-VM and an anti-VM signature, about 600 or so genes, and then sort of did gene set enrichment analysis. And what was actually quite comforting is that, of the sort of hallmark gene sets, he found unfolded protein response, which is exactly what you should find when you manipulate ER stress response. But what was definitely more interesting for us is that this ER stress-driven signature it very strongly discriminates those subclonal populations that can do vascular mimicry from those that can't. So what's important about having a signature is that we can then use those gene sets to probe other people's gene expression data. And just to kind of finish up this section, it's been thought for a long time, since the discovery of vascular mimicry, that VM is actually one of the underlying drivers of the failure of anti-angiogenic therapy. This has been hypothesized for a decade, but there's never been a shred of evidence to say that it was true. And so now with a VM signature, we can start to probe patient data sets where people, which are at least will start to let us investigate this sort of therapeutic question. Now, a couple of years ago, there was a very small clinical trial done, again on breast cancer patients, which were treated with an anti-angiogenic called bevacizumab, agonist of the VEGF pathway. And they took pre-treatment and post-treatment biopsies, segregated patients by whether they were responders or non-responders. And if we simply take the genes that showed increased expression in the bevacizumab non-responders, our pro-VM signature strongly enriches that gene set. And so we think this is, again, this is an indication, it's not a conclusion, but it's, I think, a strong pointer that the kinds of processes that we're studying have the potential to impact response to anti-angiogenic therapy in vivo. And I didn't have time to talk about this, but in addition to this secretory program, Ian has also found a master endothelial transcription factor, FOXC2, which again is also a driver of vascular mimicry. We think it operates in concert with the secretory program. And if he looks at FOXC2 driven changes in gene expression, it also identifies the bevacizumab non-responders. So I think what I've tried to convince you of so far is that we can use this, admittedly very artificial model, right? An in vitro cell line that's been cultured for 20 years. Obviously, the ones we used weren't continuously cultured for 20 years. But, cultured in a way that wouldn't select any of these properties that were relevant for in vivo tumorigenesis to reveal, I think, very new aspects of the process of tumor initiation and progression that are going to be relevant to patients. And I should say, by the way, that these properties are not stochastic. So if we take these clonal populations in this heterogeneity model, we can passage them for 40 passages in vitro, mix them back together, and these properties, for example, CTC proficiency, are dead stable. Now, we don't know whether the underlying causes of this are genetic. We've done some sequencing. We don't see any smoking guns. But, at least at this stage, we're entertaining the possibilities that these stable properties are either genetically or epigenetically, using the term completely incorrectly, determined. And so, of course, in sort of future directions for this, we really want to understand the detailed mechanisms that drive this epithelial to endothelial transition. And right now, we've just begun, because it took like a year to do an MTA with Genentech, the treatment trials to ask whether the sort of genetic or pharmacological manipulations of VM can actually modify responses to bevacizumab in mouse model trials. So that's what we know about this group of subclones that have this preferential ability to contribute to circulating tumor cell populations in the blood. We then sort of focused on what was different about this group, right? So a subset of those cells that contribute to CTC populations, only a subset of those are capable of colonizing and forming metastases at secondary sites. And so, again, we sort of undertook a similar sort of approach to finding the drivers of that pro-metastatic capacity. We did differential gene expression. We found about 100 and some odd genes that were differentially expressed between this metastasis-proficient clone and then this clone, which formed CTCs but not METs. And so that was a lot to deal with. So we decided to interpose a filter to kind of prioritize genes, which was a focused in vivo and in vitro sort of two-arm RNAi screen. We created a set of RNAi libraries with 6 hairpins against every differentially expressed gene. In vivo, we looked for hairpins that inhibited the appearance of cells in metastases in lung. And in vitro, we looked at hairpins that inhibited the ability of cells to migrate through a semi-solid matrix to invade matrigel. And then we took the outcome of those 2 screens, we overlapped them. We then asked which of the genes that emerged from these screens showed a correlation with outcome in patients, and we ended up with this, asparagine synthetase. And so I thought, well, I had avoided cancer metabolism for about 25 years. I might have to get my Lubert Stryer out again and relearn biochemistry. But so what does asparagine synthetase do? Well, basically what it does is it takes aspartate, it steals an ammonia from glutamine, and makes asparagine. And so, there were a number of possible explanations for this. People suggested that we were affecting the glutamine-glutamate ratio. We did mass spec, that wasn't true. We tested a number of other possibilities, but we really settled on the notion that we would test the possibility that it's really the bioavailability of asparagine per se that drives the metastatic phenotype. Okay? So this is just, in some ways, a recapitulation of what we saw on the screen. So 2 hairpins against asparagine synthetase, control hairpin. This is 4T1 cells, quantification of invasion into matrigel, and if we knock down the endogenous biosynthetic capacity for asparagine, we greatly reduce invasive potential. So ASNS is important for invasive capacity in vitro, but also is important for metastasis in vivo. And so here we're looking at, again, the same hairpins. We knock down asparagine synthetase. We don't see a big impact on growth at the primary site. We think we know why that is, because extracellular asparagine is relatively abundant in the mouse mammary gland. But if we look at either abundance in circulating tumor cell populations or at lung metastases, we see that knockdown of ASNS greatly reduces these. So again, we thought, all right, ASNS is important. Our hypothesis was really that it's bioavailability of asparagine. So can we manipulate these processes by changing the availability of asparagine per se in other ways? Right? So the easiest thing was just to manipulate the composition of media in vitro. And so if we take 4T1 cells and we simply increase the concentration of extracellular asparagine, they become more invasive. This is even more impressive if we do it in other models. So if we add asparagine, extra asparagine to the media of MDA-MB231 cells, they become incredibly invasive. And this correlates with reductions this correlates with changes in the amount of intracellular asparagine. We can manipulate that either by changing the media, or I'm just showing you here reduction in intracellular asparagine in media if we do AS&S knockdowns. So this is not the first time that asparagine availability has been linked to cancer, and in fact, ALL patients for decades have been treated with a bacterial enzyme called L-asparaginase. And I think this is an interesting story in target validation. So those of you who work in cancer biology know what it takes now to credential a target so that a pharmaceutical company will start a clinical trial. Right? In this case, the target validation happened when Earl was formulating tissue culture media for the first time. And he was leaving out one amino acid at a time, and he found out that leukemia cells were highly dependent on asparagine, and that was the preclinical validation for this treatment. Right? Today we have to go a little bit further. But what was important about this is that this gave us a way to go in and manipulate asparagine levels without having to genetically manipulate AS&S. And so if we simply take mice and we treat them with L-asparaginase, again, we see reductions in, this is quantification of asparagine in the asparaginase-treated animals. It really crashes in serum. And this is where we think really the gating step happens. But we do see reductions in asparagine availability both at the primary and the secondary site, and that'll be important later. They're small changes, but they're enough to be biologically significant, and I'll tell you why in a minute. But what was important for this is if we just treat animals with L-asparaginase, we greatly reduce lung metastases, and we can further reduce that if we not only restrict the availability of extracellular asparagine, but also reduce the biosynthetic capacity of the tumor cells. And so, we can manipulate asparagine in a couple of different ways here, and actually pleating it by introducing asparaginase into animals, but we can also manipulate the bioavailability of asparagine just by changing the diet of the animals. And so this is an experiment that actually sort of blew me away, but if we look at the ability of this model to metastasize, just by changing levels of dietary asparagine, we can, so this is the normal level, we can either reduce or increase metastasis in this model. And this can be further affected, again, if we restrict intracellular biosynthetic capacity. And this is just the stains of lungs. And even though we do see metastases in this low asparagine diet, they're actually much smaller and much less prominent than the ones that we see when we place mice on a high asparagine diet. I'm not saying to stop eating asparagus, which is actually what asparagine is named for, but it does raise interesting potential for clinical intervention just by changing a patient's diet in concert with their standard of care therapy. So how does this work? I don't think we fully understand how manipulating asparagine works in this model, but I think we have at least one or two really important clues. And so the first came from some proteomic, whole shotgun proteomic experiments that we did. And if we looked at those proteins which showed the greatest reduction in protein-level expression when we restricted asparagine, we found that they were annotated as proteins that were increased in their expression upon the epithelial to mesenchymal transition. And so pro-EMT proteins both decrease in their expression when we restrict asparagine. And I've just shown you the knockdown experiments here, but this is also true if we restrict asparagine availability in any other way. Right? But what's interesting about this is that this group of proteins actually shows a genome-wide enrichment for asparagine residues. And so these asparagine-enriched proteins seem to be particularly sensitive to depletion of that amino acid. And in fact, if we looked at the EMT-promoting proteins, proteins that are up during EMT, I should have written that differently, asparagine is the most enriched amino acid in this group. And I'll tell you, I don't have time to show you, that this is true in placental mammals. It's actually true in mammals as a whole, and particularly true in placental mammals. The non-placental mammals, EMT-up proteins, are asparagine enriched, but not as much as they are in placental mammals. And I'd be happy to chat with anybody afterwards if they want to talk about why I think that is. And so we think that what happens is by changing the extracellular or just general asparagine bioavailability, we're affecting the regulation of proteins that promote the epithelial to mesenchymal transition and making cells more epithelial. And we can show that in a number of different ways, but I'm just going to show you this one example is because I think it makes an important point. And so here we're staining for sort of two canonical markers, one of more mesenchymal cells and one of more epithelial cells. And if we look at tumors which we've made with either control or the ASNS knockdown, we see an accumulation of epithelial-like cells. And this is the fraction of epithelial-like cells. And here we're looking at a sort of micrometastasis at the secondary site. And remember, asparagine didn't shift all that much at the secondary site, but we still see, even with that small change in asparagine availability, a tip in the balance of more epithelial versus more mesenchymal cells. And the reason why I think this is important is because there's a vast literature, some of which may even be true, that suggests that mesenchymal cells are more resistant to all kinds of therapies, ranging from cytotoxic chemotherapy even to the more recently immunotherapy. So epithelial to mesenchymal transition seems to be important, regulated by asparagine in our mouse model. What about patients? And if we go back and we look at ASNS expression as a proxy for asparagine biosynthetic capacity, we find that high ASNS expression, we've just stratified this. Green is low, gray is medium, and blue is high ASNS expression in a number of different, these are breast cancer data sets, MD Anderson, Metabrick, or TCGA. In all cases, high ASNS expression is a marker of poor outcome. And in fact, this is not just true in breast cancers. Increased ASNS expression is a marker of poor outcome if we consider all tumor types. And there are particular tumor types where this is particularly obvious. Head and neck, squamous head and neck, and renal clear cell are just two examples where high ASNS expression is again a poor prognostic marker. Now, I just want to finish this bit by raising one point. So remember, this clone makes metastases first by undergoing an epithelial to endothelial-like transition to form vascular mimics, and then somewhere it must undergo an either an endothelial to epithelial to mesenchymal or endothelial to mesenchymal transition, right, which is gated by this asparagine-regulated process. But the point here is that I think what we're seeing here is subclones that show incredible plasticity, right, that they can become epithelial cells, endothelial cells, mesenchymal cells, and those are the populations, at least in this tumor model, that are the ones that will seed later lethal metastases. And, though I'm not a huge fan of this sort of cancer stem cell model in lots of different solid tumors, I do think that if you think about stem cell phenotypes as plasticity, this is certainly an example where plasticity is at least correlated with the ability to promote disease progression. So I just want to finish with something that's kind of fun. And so what I've told you is, sort of I thought about, since it's close to Christmas, we do like Ghost of Christmas Past, Ghost of Christmas Present, and Ghost of Christmas Future. So Ghost of Christmas Past is sort of like, let's sequence all the cells from tumors. Ghost of Christmas Present is sort of like, well, let's figure out what that heterogeneity means. And so I want to talk about Ghost of Christmas Future, which is, I think, where at least our studies of tumor heterogeneity are headed. And so this started in a way when I moved to the UK, and probably just a few months after I moved, I saw this, and that caught my attention. And what particularly caught my attention was this line right here. And I thought, well, this is a rare opportunity to really dream big. And I don't know how much any of you know about the CRUK Grant Challenge, but CRUK a number of years ago sort of adopted this strategy of promoting innovation. And they sort of did it at two ends of the spectrum. They have something called the Pioneer Award, which I run, which is $200K for 2 years. It's sort of seed projects. But they also went all the way to the other end, and said, let's fund really big ideas, really big innovative ideas. And so they went through this whole process of polling people throughout the UK, and then they assembled a bunch of ideas, and they filtered these through committee after committee, and then they delivered these to a blue ribbon international panel. And I'm not really sure what that means because I never saw them wear any blue ribbons, but that blue ribbon international panel crafted 8 challenges and essentially invited the scientific community to respond to those 8 challenges. And the one that we responded to was challenge number 5, which they sort of titled as Tumors in 3D. And so this is, sort of a Rick Klausner-esque summary of that. And so what the committee said was, what we really need is a Google Earth-like view of tumors, where you can see everything in space and interrogate each cell in a tumor and understand what it is about its spatial environment that influences its behavior. And as I started to think about this, I thought actually Google Earth is not a good metaphor, right? Because the Earth is effectively 2-dimensional. I mean, I know most of you are 3-dimensional, but if you think about it, everything happens on the surface, relatively close to the surface of this sphere, right? And so what we're talking about is, I think a much more complicated problem, not just because, tumors are truly 3-dimensional objects, but because the amount of information that we want to collect on every individual in that tumor is much greater. We'd like to have thousands to tens of thousands of data points. And so we sort of came up with this modest goal, which is to take tumors, annotate each cell in that tumor in a spatially defined way with respect to their gene expression profiles. We want to be able to monitor protein expression, modifications, phosphorylation, etc. We want to understand the distribution of mutational status within a tumor, also simultaneously map the lineage of each cell, and to do this all on a single instrument. Okay, so yeah, easy, I think. But so it is a 7-year project. So I initially thought, well, I'm in Cambridge now. All I have to do is walk down the street, and I'll find every colleague I need to find all of the expertise to do this. Turned out that wasn't true. Over the year that it took to prepare the application, this turned into a large international collaboration with 11 different groups. We've sort of structured this with a core in Cambridge. So there's 5 PIs in Cambridge, which represent math, astronomy, sort of clinical translation, chemistry, and me, whatever I do. But then we also have, gathered a set of, really outstanding collaborators, most of whom have developed some of the core technologies that we want to incorporate into this. So we have Ed Boyden, who invented expansion microscopy, Xiaowei Zhuang in the non-real Cambridge, who developed multiplex single-molecule FISH, Sam Aparicio in Vancouver, who is a real expert in sort of single-cell analysis, both transcriptome and whole genome sequencing. Joanna Joyce in Lausanne is an expert in tumor microenvironment, so helping us sort of think about how we're going to interpret these models. And Baird Bodenmiller developed imaging mass cytometry. And then all of us got together and we recruited a video game designer and started a company called Sool in Dublin to sort of do the sort of the human information interface for this. And I'll tell you about that just at the end. And so I'm just going to give you a couple of kind of fun, there's no science in this is just fun stuff. I'm just going to give you kind of a couple of fun things for sort of where we are and how we're proceeding in this path. And so the first step is to generate a real 3-dimensional model of an unperturbed tumor. And there's lots of ways to do that these days, light sheet, light field, and things. But we've actually opted for serial block face imaging, because this actually not only gives us a picture of the tumor, but then gives us physical sections, which we can then molecularly annotate later. And we take advantage of this instrument, which is called a serial 2-photon tomography microscope. It's actually used mostly in neuroscience. And this I stole from the Allen Brain Atlas. But basically what this thing does is it just hooks a vibrotem to a 2-photon microscope. And so this is a mouse brain. It's actually about the size of an average breast cancer that comes into the Addenbrooke's Hospital in Cambridge. But we basically take serial 100 micron optical and 25 to 50 micron physical sections. Now, and then reconstruct that into a 3D image. Now, what happens now is those pieces just float off into the buffer tank, right? But we've worked over the last year with the company who built this microscope to come up with essentially a collection instrument that picks up each one of those slices as it comes off the vibrotome and deposits on a microscope slide. So we can have not only the 3D image, but also the index sections from the entire tumor. And so this is just an example of an STPT this is a breast cancer PDX from Carlos Caldes's lab. The tumor cells are labeled in green. I just want to show you this to give you an idea of the sort of rough resolution of this. And so the green are the tumor cell nuclei, the black parts of the mouse stroma, and so you've seen a view of the whole tumor. These dots around the outside are fiducial marks, and I can talk about that later if people are interested in how we're going to realign all this data. But you can zoom in and really anywhere in 3 dimensions, you can see this structure with really quite high resolution. And so the idea is to take these models and working with our colleagues Nick Walton in astronomy at Cambridge, take advantage of their image processing expertise, identify each cell within the model, give it a unique coordinate, trace its dimensions, and effectively turn it into an object to which we can assign molecular characteristics. Actually, the hardest part of this is cell segmentation. Again, we haven't totally solved that problem yet, but if people are interested, I'd be happy to talk to them about that later. So how are we going to create the molecular annotation piece of this? We have a few different methods. Sort of our workhorse that we're trying to develop at the moment is multiplex single molecule FISH, built off a platform that Xiaowei Shuang at Harvard developed called MERFISH. And essentially what this is a method that takes an RNA and carpets it with about 100 single molecule FISH probes, each of which have these 2 little readouts. And these little readouts allow that hybridized mRNA to flash a digital code that tells you its identity. And so basically what you do is you take cells in which you've hybridized this probe library, and then you hybridize these individual bit readout probes, image it at diffraction-limited resolution, so that each single RNA molecule, through multiple cycles of hybridization, flashes a series of 1s and 0s that tells you what it is. And basically, it's roughly 16 cycles of hybridization to decode about 140 mRNAs. We've done up to 1,000. There's certainly a path to well, maybe not whole transcriptome, 4,000, 5,000, 6,000 genes, meaningful transcriptome, let's call it. And essentially, in each cycle of hybridization, you read a bit and then you erase it chemically. Now, I'm just going to show you an example of this in 1 color, but we can now do this in 4 colors, so we can read 4 bits per hybridization cycle. And so what you'll notice, so you see each individual cell, and this is just 7 cycles of hybridization and clearing, and you just see the bits flashing in each cycle. And what this should remind you of a lot is sequencing by synthesis, right? So if you ever saw a movie of Illumina sequencing, right, it looks a little bit like this, except not spatially defined in the way this is. Now, the reason we're pushing MERFISH and why I think this is important and interesting is that although we're reading mRNA signals, we can actually use this approach to read any kind of information that we can translate into a nucleic acid-based signal. So if we tag an antibody with an oligonucleotide, we can read it this way. So we can simultaneously read, for example, the phosphorylation state of a bunch of proteins and expression states. Using ligation-dependent FISH methods, we can read single point mutational status, which can then let us look at mutational distributions within tumors, but also lineages, because if we establish prior information from single-cell sequencing, we can reconstruct lineages using selected sets of SNVs. And so this is kind of our, at least, first stage strategy for translating that ambitious vision that I showed you on the first slide into something that looks like reality. So the idea is that this is going to produce a whopping shitload of information. And, as humans, we struggle to interact even with actually much simpler data sets. And so what we've been thinking about is, what are ways that we can conceptualize and interact with this sort of amount of data. And, if you have a piece of paper, the only way you can put more information on that piece of paper is to write smaller. We all now went from like little tiny screens to 2 giant screens. But again, this is limited. And so what we've sort of adopted, and this is where our game designer comes in, is the concept of trying to use virtual reality to sort of probe and analyze these data sets. And so over the last year, we've developed a couple of beta versions. We actually have now a fully functional piece of VR software into which we can put these gene expression data sets. What I'm going to show you is a toy data set. We're actually putting our first real data set into this now, and it should actually be ready in January. So if this were a month later, I'd be showing you real data. This is a very intuitive system, right? Because these guys are game designers. They know how to build things that people can use. This is my PA, Nikki, who doesn't have any scientific training, but she has 5 children, so that qualifies her for doing this. And so what you see is, you'll see her, in the back you see what she's seeing, right? So I wish we had the 3D version of this, but basically she can take a tumor sample, and she can grab that and she can drop that on sort of an analysis table. And this is a model with 10,000 cells. We have one with 100,000 cells. She can adjust things like cell size, the size of the model. She can map onto this. So right now we're representing cell types by icons, because that communicates another level of information. But we can also show meshes that show their actual shape. We can toggle between those. We can turn different cell types, different colors. That, I think, she just did endothelial cells, so you can see a vessel running through the tumor. This is what we call Superman mode. So you can actually enter and fly through the tumor, and see there's the endothelial cells there. And the other icons represent different kinds of cells, macrophage, tumor cells, etc. She can also map differential gene expression on this. She can represent gene expression, log, linear, whatever you want to do. And so we also have built into this, though we don't have that in this model, the capacity to identify cell lineages, and then to perform relatively complicated analyses. So, for example, we can do a t-SNE on this. We can then drop into a room which displays either, if you do PCA, you can do 3 dimensions, t-SNE 2 dimensions. We can select a set of cells, color them, pop back into the 3-dimensional model, and then see where they are. And obviously, there's going to have to be a lot of development under this, looking at, for example, things like co-expression data based on spatial distributions, etc. This is really just the very beginning. I'll stop there, just by pointing out, I talked about the guys who did a lot of the heterogeneity stuff. This crazy-looking guy here, Dario, and Christina, Sophia, and Tatiana, Fatima are missing, and Bruno here are the guys sort of working on that, the 3D tumor project. I'd be happy to take any questions.
Wow. That's quite amazing. Any questions? I'll begin. I'll just say I was quite amazed that you found asparagine synthesis as a driver for metastasis. I wouldn't have expected that. I was just interested. So that's because it's so important for the EMT proteins. So you said placental. So in non-placental, is there another amino acid that replaces it, that is more important than asparagine?
Well, so the whole placental, non-placental mammals thing, I don't think it has anything to do with metastasis. I think that, it may have to do with, and we haven't proven this, but at least one difference between placental and non-placental mammals, is that the trophoblast cells kind of invade, and there's all this stuff that has to happen. And maybe that, is driven by, asparagine as a fitness check or something like that. Here I think, and we see this, we also see, if we do ribosome profiling, we see pausing at asparagine residues, etc. I think this selection for asparagine enrichment, is linked to the asparagine-mediated control of EMT. The why is, I think, still a question. But, I think we look at the evolutionary history of this as a way to kind of get clues.
Any other question?
Very nice talk. Thank you very much. So, do you think that the change in the metabolism is at the beginning of the tumor, or the changes in the metabolism are coming with competition of cells within the tumor?
So, let's see, what do we know about this? If we look at ASNS expression, for example, in the primary versus secondary site, there's a huge increase as you go from mammary gland to lung. So we know that a subset of clones exist in the primary that at least have the capacity to show increased ASNS expression. What we haven't strictly done is fact sorted out of the primary tumor clone T cells, for example, and ask whether ASNS expression is elevated only after they leave the primary site. What I can tell you is that it's not a property of some tiny subset of clone T, because if we put a secondary barcode in, the vast majority of cells in that clonal lineage are able to make metastases. And so how ASNS is regulated or what step in the process, we don't know, but we know that at least the vast majority of that lineage has the ability to turn it up and then make metastases.
Thank you.
Any other question?
So is it known how the CMT proteins are regulated by asparaginase? You mentioned that by the translation and transcription. Is it known what's the mechanism?
So you mean for SERPINE2 and SLIPI?
Yeah.
No, we don't know the mechanism of regulation. It is possible that they're downstream of FOXC2-driven transcriptional program, but that we don't know yet. We also don't know what their direct targets are. So SERPINE2 we assume is acting as a serine protease inhibitor, but we don't know what it's, what it's binding to.
So I was wondering, in a way, are the two halves of your talk going in different directions? I mean, the second part is about categorizing all the types of cell that occur in a tumor. On the other hand, the first half is about this earlier tumor, which 20 years ago in mice, and then it's been cultured. And then you look at which cells of the 100,000 types matter, and you find that you have to watch out for one or two. So which is really important for tumors?
They're both absolutely important. So this came up a bit at lunch with the students, and so what we're really trying to do is to work across several different levels of models. And so we have cell lines, we have mouse in mouse, we have human in mouse, and then we have human specimens. And so the idea here is that as we identify context-dependent processes that are potentially important in actual human clinical samples, the hope is that we can back that up into PDX models derived from patients with similar genotypes and similar subtypes, manipulate those processes, and at least ask whether in a model they're important. So I see us as coming at this sort of from both ends and meeting in the middle. Models are going to be imperfect. Mouse syngeneic immunocompetent models are great, but they're still not human tumors. And in fact, the mouse tumor models that we have, at least in mammary gland, are, really don't capture the diversity of human subtypes. And so we think we have limitations there. We go to PDX, we capture the diversity of human tumors, but we have no adaptive immune system. And so, I don't think there's a single perfect solution to this. I think you have to come at this from both directions and recognize that it's really the diversity of these kinds of models that you need to integrate to try to figure out which of these things are important. Yeah, I know, it's total bullshit, right? You had that oh, that's bullshit. Look at this.
He always looks like that.
Yeah.
Any other further questions? If not, then I would like to present you with the MENDEL medal.
Ah. Gosh, thank you very much.
Thank you.
Solid gold, right? Yeah, solid. Okay, excellent. Thank you.